Can you see the presentation?
Um, yes, yes.
Mm, my paper of this week. Good morning. uh, Good morning. My my paper of this week uh, is shaping the gradient driving phoretic micro swimmers. Influence of swimming speed, budget of carbonic acid and environment. Mm, the in, in this work, the authors researched about the phoretic motion relies on the slip velocity between the surface on an object and the adjacent fluid. In, in order to study the, the, the motion of the inorganic swimmers or ionic swimmers. In, the phoretic effect occur on the level of macroscopic surface, the small part, suspended in the fluid and even for molecular or ionic solids. The phoretic motions appear in the absence of the body's force. For example, electrophoresis or, or thermophoresis or diffusiophoresis are some examples of phoretic effect. In the electrophoresis case, the potential gradient is the cause of the motion of the particle. In the thermophoresis case, the temperature gradient and the diffusiophoresis concentration gradient. The, the application of the phoretic motion is wide, and additional interest in recently in mechanisms for chemotaxis effect. Uh, it, it, it is very useful in this area. Mechanism for chemiotaxis. In this work, uh, the phoretic propulsion by generate for ion exchange, and uh, the monitor swimmer propulsion are made for tracking and concentration of carbonic acid via high resolution PHD. Uh, the objective of my presentation is to focus on obtaining the experimental pH mapping in order to orient, 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 orientate my uh, laboratory work project, the pH image and control.
1 minute left.
Yes, in the focus of pH mapping. No? In this work, the general photometric approach is used in this investigation, and the image are analyzed uh, by means Python script and analyze the colors of the image, and use the Lambert-Beer law for calculate the mean absorbance A with this equation. And if they use a relative absorbance, they can to obtain the calibration absorbance ratios and to know the pH concentration. In these figures, the micro-photometric testing are made, and calibration curve displaying absorption ratio of the color for the different pH in this figure. And the range of the interest for they it is. In conclusion, the pH the pH experiment with another approach, the photometric approach, is a good idea. In our case, the pH mapping we can make with another approach. Thank you very much.
Thank you, Luis. Eric, can you present?
Okay, I'm set. I'm going to share my presentation. Can you clearly see my screen?
Yes. Yes, Eric.
Thank you for your confirmation. So, well, good morning, everyone. Today I'm going to talk about the recent advances in the development of aerospace materials. This paper aimed to review the recent advances of major aircraft material. As a result, studies has been conducted to develop materials with superior mechanical performance and corrosion resistance to full improvement in both performance and life cycle cost. In this context, lightweight of aircraft frame and engine design with materials of improved mechanical properties can improve fuel efficiency, increase payload, and increase flare range, which directly reduce the aircraft operating costs. On the other hand, aluminum-based alloys has been dominating in aerospace material over 80 years. However, this situation has been changed in the last year. For example, here, Figure 1 provides information about the total material used in Boeing series aircraft. As you see here, aluminum-based alloys tend to decrease. Composite materials has been experienced a rapid increase in total materials in the latest Boeing models. This is mainly due to their higher specific strength and better corrosion and fatigue resistant than most metals. Regarding the design criteria of the aerospace materials, it depends on design requirement of each component, including loading condition, geometric limits, environment aspect, and maintainability. About the design criteria of the airframe materials, they should be designed to provide long-term support for both the static weight of aircraft and additional load subjected from service. On the other hand, regarding design criteria of the aircraft engine materials, they are required to possess low densities for weight reduction and good mechanical properties in high temperature and corrosive environment. In this context, titanium-based alloys, aluminum-based alloys, and polymer composites are material choice for this application. Now we're going to talk about the recent advances in the aircraft materials. Aluminum-based alloys still remain an important frame structure material. So we can see the recent research on the development 2000 and 7000 series aluminum-based alloy for aircraft application, such as 2000 series aluminum copper-based alloys, which possess superior damage tolerance and better fatigue resistance than the other series aluminum-based alloys. Furthermore, we also have a 7000 series aluminum zinc based alloys, as well as aluminum lithium based alloys. At the same time, we could mention that the use of titanium based alloys has been increased in recent years. As shown in figure 3, the operating engine weight has increasing 1%, has increasing use of titanium-based alloy from 1% to 90%.
1 minute left.
Okay. Regarding composite materials, their use has greatly increased, as we can see in figure 5. Composites constitute more than 25% of the Airbus 380 and 50% of the Boeing 787. Okay. As a final conclusion, we could say that the significant progress has been made in the development of both aircraft structure and engine materials. Furthermore, the design criteria for the aircraft structure require the material to possess appropriate mechanical properties with suitable damage tolerance under different conditions. That's all. Thank you for your attention.
Thank you, Eric. Okay. Now I will talking about this paper called the dialogue between protozoa and bacteria in a microfluidic device. In nature, protozoa play an important role in controlling bacterial populations. This article proposed a microfluidic device for the study of the behavior change of protozoa due to their chemotactic response in the presence of bacterial cells. In addition, the volatile organic compounds, or VOCs, produced by bacteria, in this case Listeria, were identified by mass spectrometry to determine their actions against Eulina. The device was made with nitrocellulose and consists of 3 channels that were made, you can see in this figure, that were made with a laser cutter, and the membrane was interposed between 2 glass slates. The central channel includes 6 dead-end side chambers, 3 in the upper side, and 3 in the bottom. The lateral channels are for the buffer and for the entrance of the bacterial samples. And for testing, a suspension of euglena was allowed to flow through the central channel in the volume of 100 microliters. The euglena suspension was added to the central channel, and the flow rate was controlled by a syringe pump here. The system was equilibrated for 20 minutes and time zero was before the introduction of the chemofactor into the lower channels of the device. The videos were taken at intervals of 10 minutes up to 60 minutes to determine change of distribution of euglena cells. In the results, you can see in this image from 0 to 30 minutes in the top and bottom chambers. Although there are not chemoeffectors, more euglena cells reach the top sides here of the chambers, and the cells gradually increase over time. And exists a greater number of cells in the upper chambers, in the 3 chambers. Now here, for comparisons in this figure, they use polystyrene bead suspension as an unmobile control. Didn't reach the bottom of the chambers due to lack motility. More beads accumulate in the central channel directly between the entrances. This could explain why a higher concentration of euglena is observed in the top chambers. Also, these observations could be attributed to the uneven distribution of light on the microscope stage, since euglena is photosensitive. In this figure, you can see the percentage of euglena in top chambers without chemoeffectors. Here, you can see the displacement of euglena cells over time. At time zero, more than 60% of euglena was at the top, and they were rapidly
1 minute left.
Reduced after listeria was introduced into the system. Here you can see in this figure. This figure represents the reductions, the euglena reductions rate. Yeah, this population's change suggests that the chemical release by listeria attract the protozoa. Now, the small molecules produced by listeria were identified with mass spectrometry and chromatography. These compounds were performed in the same assay. Discovered that listeria VOCs are the chemical cues for euglena. Furthermore, they identified several VOCs associated with bacterial samples. Among the 4 VOCs studies, the canal showed the most significant influence to attract euglena. In conclusion, the efficacy of a microfluidic device for the quantitative evaluations of the response of protozoa to listeria has been designed and demonstrated and demonstrate. In the future studies, these devices can be used to compare the attraction of protozoa to two different species of bacteria. Thank you. Okay, Daniel, can you present?
Okay, thank you. Can you see my screen?
Yes.
Okay. I'm going to present a novel two-layer integrated microfluidic device for high-throughput yeast proteomic dynamics analysis on single cells. Basically, when we have to prove several strains of cells, we need to make different chips, different devices for different conditions or for different strains. In this case, they prove just one new design to prove several strains with different conditions in the same micro device, but with two layers. And here we can see a schematic representation for that. And in the last researches, they used a one chip for different environments. And different environments, they have two strains. But here they represent schematically when you have just one device with different inlets and different outputs in the. For n, here we have n is a number of the n number of the strains, and in the m is the number of the conditions. Can you how do you can see here we can see all the strains with all the conditions. So with immaterial methods is the procediment that we know. They use 4 masks here to make the siliceous wafer. Yeah, they use siliceous wafer with SUV polymer. They use for mask and to mask 2 masks for the conditions plate, and 2 masks for the strain for the seed layer. The method is lithography, and here we have how they use them. We see they see the yeast here, and these channels they have a little barriers because they did 2 repetitions by the several conditions, but for the conditions, and then they fill the chambers with the different conditions here. In this case, 1, 2, 3, 4, 5, 6 conditions. What were the conditions? The conditions were different concentrations of glucose, because when you give I can't remember the word, but when you have different concentrations of a lower concentration of glucose, you you stress the yeast, and then they
1 minute left.
express a different. Okay, thank you. A different markers. In this case, GFP attached from several genes. Here we can see the medial inlets and the chambers, the little barriers to do two repetitions for to everyone. And here we can see all the response of the yeast. Well, here we can see what was the process to obtain the readiness of the GFP attached from every gene, and they were compared with another work with previous works. So the discussion and conclusion is that this design is a functional design. They can use several strains in several conditions, and just in one device. In comparison with another with previous works, then they use 100 strains, for example, and they have a lot of time between seeds. And that's all. Thank you, everybody.
Good morning, everyone. Today, I'm going to talk about a rapid separation and identification of beer spoilage bacteria by inertial microfluidics and MALDI-TOF mass spectrometry. Okay, so the idea of this study is to separate the microorganisms that can spoilage the beer from the yeast. So first, let's talk about the dimensions of the bacteria. For example, Lactobacillus febris, the diameter is between 0.3 to 3 microns. Albeit yeast diameter is between from 4 to 5 microns. So why is it important to separate bacteria from beer? So from the brewery companies, they are very worried about it because the presence of bacteria or pathogenic microorganisms could affect factors as high ethanol content, high acidity, low dissolved oxygen content, and high carbon dioxide content, and also nutrient availability. So here, these authors propose to create a microfluidic chip that consists in a PDMS body and a PDMS layer, and they are going to separate through this spiral design the bacteria from the yeast. So as you can appreciate here, the yeast is represented in this scheme in green, and the Lactobacillus brevis is represented in pink. Okay, so they use this syringe pump, and then if we see the channel, the transversal section, it has a trapezoidal shape. As you can see here, the AA transversal cut, so you can see the dimensions. So the inner wall, the diameter of the inner wall is 100 microns, while the outer wall of the channel is 40 microns, and they are separated by a distance of 400 microns. Okay, so the principle of the separation is that yeast are going to be focused along the inner wall of the channel, while the bacteria in this bifurcation point, as you can see here at the BB transversal section, the bifurcation point, the bacteria are going to be dragged. So they are going to be located close to the outer wall. And that's how they separate the bacteria from the yeast. So these particles, because they have a different size, the inertial lead force is going to influence the bigger particles, the yeast, while the drag force that you can calculate with the equation number 2, it's going to influence the location of the smaller particles, the bacteria. And as you can see here, in the drag forces, the D number is considered. Okay, here we have a image of the yeast and also the Lactobacillus brevis. First, they optimize the flow rates using the matrix was PBS, and then they are going to use the matrix beer. But let's see what happened with the PBS. So what they measured was the focusing efficiency. So let's see, the focusing efficiency is the percentage of cells existing in the inner and outer outlet divided by the total number of cells existing in the device. So first they tried these 3 flow rates, and they saw that the flow rate of 1.5 milliliters per minute was the one that gave them the best focusing efficiency for
1 minute left.
this yeast. Thank you. And so they optimized that flow rate with the PBS and also with the beer. And then after they separate the particles, they use this spectroscopy to analyze the species and to identify the species and genus of these cells. So, and they also said that it's very cost efficient because each chip costs $4 to prepare, and then each assay of this spectroscopy, after you buy the equipment, it costs $1. So it would be by $5 you can have you can identify the species. Okay, thank you.
Okay. In this presentation, the topic Fluorescence-based sorting of C. elegans via acoustofluidics. Different phenotypes are important for more research on worms, especially in genetics. Here are chips mentioned, capable of identifying worms of interest based on the expression of fluorescent protein. The chip can classify worms at different stages of development. Worms in L3 and L4, about 70 L3 worms and 115 L4 worms are identified per minute. Okay, in this part, in figure 1a, illustration of the complete system design and operation to classify worms include 3 parts: optical detection, execution, classification of worms. In figure 1b, consists of a single player of PDMS with 2 inputs to load the C. elegans samples into the buffer. It also has 2 outputs to download green Florence protein and wild-type worms. In this part, in IDT, they are interdigital transducers deposit in lithium novate, some substrate used. During the process, the green fluorescence protein expressing wild-type worms are loaded into the microchannel. Each worm enters the detection window where two optical fibers distinguish the worm genotypes. If a worm is identified with green fluorescence protein, the IDT is activated to generate soap. The worm is pushed to the desired exit. Otherwise, the worm will flow towards the exit of the wild worm. Okay, in this part, in figure 2a, after injecting into the microchannel, the worm passed through a gradually narrowing channel and then hit to the optical detection window. The channel has 16 micrometers wide. It serves for the passage of L3 and L4. In addition, the smaller distance between the worms and the fibers can increase the detection accuracy. The schemes indicate when the worm expressed green fluorescence protein pass through the optic region. Fluorescent light is detected in the optic region. Okay, bueno, in this part, figure 2b, a wild type L3 worm, and in figure 2c, L3 worm expressing a green fluorescence protein through the optic region. Okay, and this part, okay, in this part, figure 5a and d show results of a single experiment were photographed with a fluorescence microscope to perform the yield and purity calculation. Almost all worms from the green fluorescence protein expressing outputs showed green fluorescence, while most wild-type worms did not
1 minute percent.
show green fluorescence. And this part, figure 5E and F, purity and yield shown separately. In L3 worms, purity and yield decrease with increasing throughput, remaining above the 19% of yield with 100 worms per minute, and 80% with 155 worms per minute. For worm cell 4 is purity and yield were less than L3. Their data indicate the same thing. In conclusion, a chip has been developed to classify worms that express green fluorescence protein in a fast and biocompatible way. Thanks.
Good morning, everybody. I'm the next. Okay. I'm going to novel nature-inspired algorithm for optimization. In this paper, the author presents a new or a novel algorithm based on Squirrel search. In many problems of engineering, the optimization is a principal component because this is the best form to compute the parameters of another problem. So in this paper, the author presents an algorithm based on the behavior of a squirrel in order to search food. The optimization problems are aborded in different ways, and one of them is methods of inspired in nature, and in general are called as evolutionary algorithms. So this method can apply it in different applications, for example, biomedical signal, image processing, flexibly job shop, and another problems. The principal components of these algorithms are to has two stochastic components in order to improve the time to compute the solutions. In this table, many of known algorithms based in this approach is presented, and in this case, the Squirrel behavior is the way to model mathematical in order to describe the algorithm. So four principal steps are compounding these algorithms. For example, the first is the random initialization of particles in the space of feasibly space of solutions of the optimization problem. The next is to evaluate the fitness function. The next step is to describe mathematical model to improve in the next generation the fitness function value. With the purpose is to generate iterative algorithm in order to find in the final step the best solution. Squirrel search is based on the way of squirrel find food. So in this part, the author presents the equations to use for this search. So the principal statement in this case is the way of find or search the best solution. In order to prove this algorithm, a benchmark function is used, which allow to compute the performance of these algorithms. For example, this is an optimization function used to evaluate the performance of optimization algorithms. For example, in this case, this function is in 3D space. Passive space, which is the function exists, and each iteration can compute the best solution. In this case, for example, is a minimization problem in order to find the best solution. Algorithms also are compared with another nature-based algorithms in order to present the improvements or new features. So, for example, in the case of SSE algorithm, in this image, for example, the evaluate function for SSE algorithm is improved to compare with another algorithms.
1 minute left.
Okay. So another form to evaluate these algorithms is according to the statistical measures. For example, the mean and standard deviation in order to compute the error in different functions. For example, in this image, different functions are evaluated with different algorithms, where SSE algorithm is the best optimization. Another function is presented in this image. So the complex optimization problems can be described in any dimensional space. So in this case, for example, this function is a very used function to evaluate the algorithms, where the best solution is found in center of the function. So finally, the authors evaluate the algorithm in real-time processing problem, where the temperature is used to compute the best form to maintain this temperature in control environment. For example, in this case, 4 sensors of temperature in a row has the inputs of the system, and the algorithm compute the best form to evaluate and to send signal to the actors for control temperature. Well, finally, in this paper, swarm algorithms are used to avoid optimization problems in order to improve the time and improve the solution optimization, computing processing, and time. That's all. Thank you.
Hello, everyone. I'm going to be presenting this paper, Microfluidic synthesis of robust carbon dots functionalized photonic crystals. Well, for those who don't know or don't remember what quantum dots are, particles of a few nanometers that have optical and electronic properties that differ from larger particles due to quantum mechanics. For example, they possess photoluminescence. And photonic crystals are periodic dielectric structures that are designed to form the energy band structure for photons that can either allow or forbid the propagation of electromagnetic waves of a certain frequency. So in this paper, we are going to explore an assembly strategy to combine quantum dots and photonic crystals to prepare hybrid microbits that have excellent optical properties, are heavy metal free, environmentally friendly, and have a superior performance material. Okay, so here in the first picture, you can see a schematic illustration of the synthesis. They combine mainly two elements, the carbon dots that have a blue emission and colloidal particles that possess good monospersibility and create photonic crystals that have a green structural color. So the carbon dot solution containing an amino group and the colloidal particles containing carboxyl groups are the building blocks to efficiently assemble these carbon dot functionalized photonic crystals through continuous droplet microreactors in a microfluidic assembly strategy. So the synthesized hybrid microbits show dual optical signals with bright green structural color under sunlight and strong blue fluorescence under UV light. Next, well, they characterize these microbits, but I'm going to focus a little more on the properties of the structural color, fluorescence intensity, and fluorescence stability. Well, the first thing they saw is that the reflection spectral intensity of these hybrid microbits is significantly enhanced. And they also saw that when you have carbon dots powders, aggregation will result in quenching of the fluorescence, but loading these carbon dots on the photonic crystals can effectively avoid this aggregation and the fluorescence quenching. And so these can also enhance the diffraction color by increasing reflection and reducing scattering. Then also, photonic crystals are commonly present different structural color when viewed from different angles, which can also limit their applications. So these prepared hybrid microbits show angle-independent structural color because of the spherically symmetric structure they have. So, these microbits have an angle-independent photonic bandgap showing green structural color, not only on the surface, but also in their cross-sections. And, mainly because of the carbon dots, both surface and the cross-sections of the hybrid microbit also emit bright blue fluorescence under irradiation of UV light. Then there's another interesting thing about these microbits is that their structural color and fluorescence intensity respond to different metallic ions. So the reflection spectra show different degrees of redshift. And this phenomenon may be attributed to the metallic ions changing the photonic bandgap of the photonic crystals. And the fluorescence intensity of the structural color in these microbits are responsive even when the concentration is as low as 0.1 micromolars. And these hybrid microbits
1 minute left.
Okay, can be arranged in an array to extract optical signals and apply in ionic sensing with a dual response of structural color and fluorescence, like in image D and E. And finally, they found that the film prepared by these particles not only has a more vivid structural color, but can also reduce cracking. And this brighter structural color is mainly attributed to the fact that the carbon dots loaded can finally enhance the diffraction color by increasing reflection and reducing scattering. Also, based on the blue fluorescence properties of these microbits, they applied them in what's called a light belt, and they were fixed in a UV LED chip emitting at 390 nanometers through silicon. And the light belt emits bright blue fluorescence when the switch is turned on. And because these raw materials used in the preparation of the microbits are low toxic and harmless, they can prepare fluorescent light belts that can be used in, for example, wearable bracelets, like you see here in this picture. That would be all for this paper. Thank you.
Okay, thank you. Okay. Okay, can you see my screen?
Yes, yes.
Okay. Okay, in this case, I am going to talk about this parasite that is very known. This is the Trypanosoma. In this case, Trypanosoma africans, and this is a no, this is not my paper. Sorry. I can, because I am the computer of Anita, can in order to continue with the presentations, please the next, and then I will finish.
Okay, Camilo, can you present?
Okay, yes. I'm try to present now. Please stop and change. I need of your screen, Gustavo. Can you hear me? Okay, thanks. Okay, everybody. In this case, I am talking about microfluidic protein detection and quantification using droplets morphology. It's for me, it's very interesting because it's a new method of classification of proteins. But you know, the protein is it's very important for study and the sense and other genomics expressions or different studies. Okay, but in this case, they propose a new method for with droplets and the shape and the weight classify the proteins. Okay, but the first the first figure and use an first fluid and HPLC and techniques, but after make a droplets with and microfluidics techniques. It's conventional. Or after classifies the droplets for the shape. Okay, on the weight. Okay, in the figure B, using and the pressure or an electron regulation for understand these flow rates and the protein sample. Okay. After in classifies the droplets for how these droplets or weight and form, and this occasion is for understand the droplets on form and the weight in this figure is the difference for protein concentration. That's it's they understand this the different protein depend of the protein concentration and the type protein. Okay, well after this compare that technique what's HPLC and for normal techniques, for example ELISA or physical techniques, immunoprecipitation, right? Okay, after classified for when we make droplets in the normal experiment, well, this effect that depend the flow rate. Okay, but in this case for concentration and type of protein, right? It's very interesting, but we need to understand for concentration is a different protein on the form and the shape of droplets. Okay, and this compared for other methods or common methods, right? And this for classificates are it's understand how make a droplets, but the concentration. Okay, look in the make a composition of the droplet site and interstitial tension of the flow rates. Okay, for weight for protein, right? And when the flow driven system and change this phenomenon, it's always
1 minute left.
Okay, make okay. Okay, after comparative detection method for this method of now alternative of for quantification and detect different protein. Okay, but in this case is more efficiency and method for detection protein and this limitation of study in the site and the protein displayed on the other weight protein or previously HPLC techniques. Okay, that is all. Thanks.
Okay. Sorry about the miss, that we had a misunderstanding. Okay. 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 In this case, I'm going to talk about this this technique that they use for isolate African trypanosomes. This is the one of these. This is in a review article, and this is the few of the studies that there are in the bibliography. There are not too much studies about this, especially with trypanosomes, and this is the cycle of trypanosome. This is the shapes that the parasite adopts in different fluids. For example, in salivary glands, in mid-note, that this is medium intestine, and this is in the fluid, in the bloody fluids. It has a motility. This is quite difficult to see in the normal wave. It means with the flask. That is the reason that in this case they use microfluidic devices in order to separate, especially from blood. It means we have these shapes in these studies. I will show you the studies. The first study that they use is the electrophoretic microfluidic device. You can see in the borders they use for separate for blood. In the borders, the blood is erythrocytes, and the parasites goes to the center of the microfluidic device, and they use for separate. This method is used for separate in order to collect, in order to perform the diagnostics. This is the idea to improve, because the most of these parasites, it's quite difficult to identify with the centrifuge, with these things, and this device could be a great opportunity to get the diagnostics very soon, especially in the field. I mean, in the people that really need this. Okay, then and there are another technique that called is DLD. That is a principle of separation according to the flow. I'm going to show you. Okay, this is the principle of microfluidic. When we have a flow, when we have these structures, the erythrocytes or red blood goes to the direction of the flow, but the parasite that is quite bigger, they follow this direction. And it's easy to monitorize and get also the separation. This is a technique used microfluidic flow in order to separate from erythrocytes, and also we can follow this. This is the microstructures that they use. This is the size that they use in order to separate these parasites from the red blood. Another study with the same technique, they use this kind of microfluidic in order to attract or in order to confine the parasite, and they took videos without drugs, and then they used drugs.
1 minute left.
Yeah, and then they took also pictures, also videos, and then compared how the mobility change when the parasite get a concentration or different concentration of drugs. And this is another interesting thing that it's also possible to do with the microfluidic, because this is the configuration of the chip that I told you at the beginning. They have small squares and then increasing the size and also increase the concentration, and in the middle they put the drugs. This is the techniques that they use also for separate and also for testing drugs. This is the final study, and in conclusion is easy to perform and design a microfluidic device in order to get more information of this parasite that is not studied good in microfluidic devices, and it's also an opportunity to find new solutions. Okay, thank you.
Thank you. Have you questions?
I have one. Gustavo, do you know how they load the parasites in the chambers, and which software they use to track the movement in the last experiment?
They use the microfluidic pump, but they use they confine the parasite, but when it's in the smaller size, and then when they grow, they get this size because they are looking for the specific size. They put the drugs because it means the parasite that grow inside of the microfluidic, and then they put the drugs. And with another question,
which is how they track the movement?
They use a MATLAB. They use algorithm to track with MATLAB.
Okay.
Yeah. It's not intelligent. It's only a simple track.
Okay.
Mm-hmm.
Another question? No more questions? Okay, I will ask another one while you are thinking. Rocio, do you know how long the nanoparticles keep emitting fluorescence after they are excited?
Yes. Sorry. They keep emitting because the fluorescence comes from the way they are built. As long as you have the UV light on, it keeps emitting.
But I am not an expert, but normally you have to excite with some kind of light, and then
Yes,
the particles
with
emit. And when you have them in the, I don't know how to say the collar or the pulsera, they keep emitting if
they keep emitting in the necklace because they are fused in an UV chip that you can turn on and off.
Okay. And you mentioned that they were not toxic. Are you sure? Normally, the nanoparticles are not toxic from carbon nanoparticles.
They non-toxic. They put not toxic because usually this kind of combination between quantum dots and photonic crystal are done with heavy metals. So the non-toxic part comes from not using heavy metal to create these microbeads.
Oh, okay. Well, I know more questions for another one. Okay, I will ask to Daniel. Do you know how they collect the DNA from each chamber to make the PCRs? Because it's on a small amount of sample.
For me?
Yeah.
The question was for me? No, they don't collect. They didn't don't collect the DNA. They just took a picture from the chamber because they just saw the response of each cell for the stress condition. The stress condition was the lack of glucose.
But if I remember, well, you showed at the side of the picture a pattern of gene expression, or I was wrong?
Yeah.
And how
was
they
the gene expression?
How they check the gene expression? Because it's a problem when
see my screen?
Yeah.
Okay. Well, here, the GFP was attached for an exclusive gene. So when these genes are intervening in the downstream pathways of the metabolism of the yeast, so when they lack glucose, the pathway start on or start off. That was the experiment. When they lost glucose in the medium, in this part, the MS2 protein goes for the nucleus, and that was in all these conditions. But with FP1, you can see that the protein attached with GFP exited from the nucleus, and then it was concentrated in the cytoplasm of the yeast. Then they didn't collect anything.
But from the picture B, from where they took the sample to make the gene expression?
The gene expression is the fluorescence. When you have a fluorescence and a mobile,
okay.
They use a software for yeast, an exclusive software for yeast.
Okay.
And image J, but not for this, just for measure this one. And here you can see the relationship with the fluorescence and the glucose concentration.
Okay. Okay, I will check in the article. In any case, I will ask you privately. Okay. Well, if anybody have another question, if not, we can see each other next week.
Okay.
Thank you, everyone. Bye-bye.
Okay, bye-bye.
Thank you.
Bye
See
bye.
you.
Thank you. Bye.
I'll see you.